Hello, my name is Gallagher, and thanks very much for attending my webinar today. I'm going to talk to you today about a particular new exciting initiative, Collaborative Cancer Research Centre, Breast Predict, and how we're applying this particular approaches relevant to systems medicine towards developing better treatment strategies and also new diagnostic options within the breast cancer context. So just a bit of an introduction around breast cancer, just to kind of give you some kind of context behind the centre. I'm speaking to you today from Dublin, Ireland, but you could, if you look at the stats within Ireland, they're quite similar to what's seen in the UK and the US. We have this issue now at the moment that 1 in 10 women face a chance of developing breast cancer within their lifetime. And then if you look in terms of the worldwide incidence of breast cancer, we see that the global incidence of this disease is increasing. Some of this is due to improvements in screening and also lifestyle factors. But we know that from 2012, it's predicted to increase from 1.7 million women with breast cancer up to over 2.5 million in 2035. Now, we know that the news is not all that bad because due to, I suppose, advances in screening, surgery, various different therapeutic options, including conventional classical chemotherapy, hormone therapy, but also the development of new and more exciting targeted therapies, mortality due to breast cancer is decreasing, such that we have, if patients present with localized disease, they have a very good chance of long-term survival. Now, we still have an issue with patients which present with distant metastases in terms of the poor survival rates of these patients. But in conclusion, most women diagnosed with breast cancer now survive. And this is really provides a context behind that. It's been becoming more important that we tailor treatments and also avoid some unnecessary side effects associated with treatment. And we know that research has made a significant impact in terms of our understanding of some of the risk factors associated with breast cancer. And some of these are well known both to the scientists but also to the lay public. We know that, for example, obesity, poor diet, alcohol, lack of exercise, and hormone exposure are changeable factors which can actually impact or impinge on breast cancer risk. We're also familiar with the concept of particular susceptibility genes or factors or mutations in particular genes like BRCA1 and BRCA2 and other similar genes which predispose or confer a higher risk of getting breast cancer. Obviously, as a person ages, the risk of cancer in general increases. And these factors are obviously unchangeable in the main. Alongside these knowledge about risk factors, we really have come to understand a lot about the biology of breast cancer via a variety of different approaches, sequencing approaches or various other omic approaches. For example, we know a lot about the different subtypes of breast cancer, the relative response rates to different treatments. We know we have a lot of new biomarkers which can be used in the breast cancer context. So there's a lot out there in terms of our arsenal to deal with breast cancer. And this is just captured in the next slide, where we've really charted some of the key seminal steps throughout just over the last century, approximately, or a bit longer. For example, if you look at just before the end of the 19th century, we have essentially the development of radical prostatectomy. Again, around that time period, George Beatson in Scotland performed the first ovarectomy as a treatment option, removal of the ovaries as a treatment option for breast cancer. Now, in terms of the first impact in terms of drug treatment, we can actually look at the development of tamoxifen. This drug has had a major impact in terms of particularly in an ER positive or estrogen receptor positive context in terms of targeting breast cancer. And it's probably, one would argue, one of the most successful anti-cancer drugs that has been developed to date. Again, in the mid to late 80s, we have the development of more targeted agents. Again, tamoxifen is an example of a targeted drug, but also we have, I suppose, a poster child of targeted drugs, Herceptin, which targets the epidermal growth factor receptor HER2, which is overexpressed in approximately a third of breast cancers. And these particular subgroup of breast cancers have a very poor outcome in terms of their treatment response. And due to the development of these, what are called HER2 targeting drugs like Herceptin, their actual outcome of patients with this type of subtype of breast cancer has dramatically improved in recent years. Again, when we look at the impact of the omic era on breast cancer, it has had made a significant change in terms of how we actually look at breast cancer. We have these classical subtypes of breast cancer, which can be divided into estrogen receptor positive breast cancer, triple negative breast cancer, which are devoid of the estrogen receptor protein, progesterone receptor, and HER2, and then HER2 positive breast cancer, where they overexpress this HER2 protein. And again, during the 90s and also in the early 2000s, there was a lot of work really on defining new molecular subtypes of breast cancer via gene expression profiling. And some of these discoveries have now been translated into new diagnostic tools which are used clinically, for example, like Oncotype-DX and Mammoprint. Alongside that, we've had a development of lots of other targeted agents. For example, we have agents which are targeting particular kinases, etc. Now, when we look at the, I suppose, the different types of agents which are used to treat breast cancer, still within the, I suppose, the arsenal in terms of dealing with breast cancer, we have classical chemotherapy. Generally, when we talk about targeted agents, they're used in combination with classical chemotherapy. What I just indicated here is really just a kind of various different agents which can be used to target the HER2 protein. This could be the prototypical monoclonal antibody trastuzumab or Herceptin, and then also you have these variety of other agents like TDM1, which is this new antibody drug conjugate, or you have low molecular weight compounds, not an antibody, such as lapatinib, which can actually inhibit the tyrosine kinase activity of this receptor. Also, as I indicated before, that the sorry, 1 second here. Yeah, also as I've indicated before, hormone therapy plays a very important role in terms of the treatment of breast cancer, and we have a variety of different agents, including the kind of, I suppose, the standard bare tamoxifen, but we also have aromatase inhibitors which can be used to deplete the supply of estrogens systemically. Now, alongside these very widely used agents, we have also new agents starting to be approved, targeted agents. For example, everolimus, which is an mTOR inhibitor, has recently been approved for the treatment of breast cancer, particularly breast cancers which essentially are resistant or have had relapse on endocrine therapy. There was an approval by the FDA for the use of Avastin for breast cancer, which has been rescinded recently because of lack of efficacy. But again, there is still an interest potentially in the use of angiogenesis inhibitors to combat breast cancer. Again, what I've indicated at the bottom of this slide is really there's a whole slew of agents out there in clinical development, really covering a variety of different molecular targets which are being tested in breast cancer. And some of these, for example, one would assume will be approved over the coming years. I've just highlighted a couple of these. For example, there's a big interest now out in agents which can actually target CDK4 and 6, like this agent from Pfizer, palbociclib. There's also a big interest really in agents which can actually target defects in DNA repair, such as the PARP inhibitors. So I've kind of painted, I suppose, a brief picture about the kind of background behind breast cancer, and I suppose it's important to describe really why do we need a collaborative cancer research centre. And so an important document that might be of use to you if you're interested in the breast cancer arena is a gap analysis that was performed by the UK charity, the Breast Cancer Campaign, and they brought together just under 80 to 100 investigators with specialist expertise in either oncology, basic science, translational research, with an interest in breast cancer, really to look at the critical gaps and translational priorities required or important issues in terms of that are still outstanding in the context of breast cancer. And some of the conclusions from this document, which was published in Breast Cancer Research in 2003, was that we need to really improve our resources towards breast cancer research. We need to develop a fully cohesive and collaborative infrastructure to support research. We need access to appropriate and well-annotated clinical material. We need to encourage collaboration between researchers in different scientific fields, and also improve clinical trial methods to better meet the complexity of modern treatment options for patient benefit. So this leads into this initiative which I direct called Breast Predict. And this program is funded by a major charity within Ireland called the Irish Cancer Society. It's the main charity which funds cancer research. And it's the first of what they term a collaborative cancer research centre. It's a virtual centre where we bring together essentially researchers from multiple different institutions around Ireland, plus a cooperative oncology research group that is carrying out clinical trials activity. And really to work in a collaborative research environment is not really about bricks and mortar, it's about the individuals working together in a collaborative team. So this program kicked off in October of last year, and we have funding really for a 5-year program. We've just completed the first year of this program, and I'm going to give you some highlights from our first year. It's the first of these new collaborative cancer research centres which are going to be funded by the Irish Cancer Society over the coming years. This particular one is focused on breast cancer. The other subsequent centres might be focused on different cancers from different origins, or maybe focus on a mechanism of disease. And really the goal behind these centres is really to bring, and this one in particular, is to bring together cancer researchers, and we obviously have an interest in researchers with an interest in breast cancer from various different disciplines around Ireland, with the common goal of improving the outlook of breast cancer patients, both in Ireland and worldwide. And so this centre has 3 main themes or components. There's a translational research component where we, I suppose, come up with new discoveries from the bench, try to bring these forward and look at a pathophysiological relevance in clinical tissues, for example. There's also a clinical trial component or intersection with a clinical trial component. And then another important perspective is also take on board information at a population-based level. And again, our programs are structured such that we also have intersection across these thematic areas. I suppose if we look at a collaborative centre, why we would do it and where the context behind it is really, again, we want to bring together a group of talented researchers to tackle a number of key issues relating to breast cancer. We want to really maximize use of the resources. There's no point in us, for example, or multiple different institutions really, for example, buying the same piece of equipment, but wasting money on multiple pieces of equipment, but not really maximizing those resources. So really, we want to encourage researchers to share those resources and expertise. We're really needs driven. We really want to focus on the needs of patients. There's also a training mission within the centre where we organise various different seminars and workshops. And again, we want to really bring together specialists, for example, particularly in the area of computational biology, bioinformatics, mathematical modelling, in conjunction with the practitioners, be they surgeons, oncologists, pathologists, and really kind of maximise the interdisciplinarity behind our centre. As I mentioned, avoid this duplication of resources. So who are we? We're essentially a group of 20 investigators dotted around Ireland. In the background here, it's a map, loosely, of Ireland. But what I'm just indicating here are the 10 PIs which are involved, and then on the right-hand side are just a list of some of the other funded investigators. In total, we are 20 investigators from 6 academic institutions around Ireland, and that's indicated on the bottom right. I won't go into detail. You can actually look at our website in terms of what are the institutions. But I want to highlight one particular partner. In addition to these 6 academic institutions, which are primarily the main universities and other academic institutions in Ireland, there's also a important cooperative group, ICORG, which performs clinical trials in the oncology space across both the Republic of Ireland and Northern Ireland. And really all of the practicing oncologists, be they medical oncologists, surgeons, really have signed up to an integrated program for delivering new therapies to cancer patients within Ireland. And so our program really leverages that important resource. So just in a bit more detail about the research that's taking place within the program. And just to set the context, we know that breast cancer is not just one disease. We know that, for example, patients, if they may present with similar clinical features. They could, for example, 2 patients could be ER positive, HER2 negative, they both may receive hormone therapy and chemotherapy. But we know that, for example, those patients can diverge in terms of their response, so patient A and patient B. And this is indicated here in this scanning concept, where we have two patients which obviously have diverged in terms of their treatment outcome. We also have this issue of differences between, for example, a primary tumor that may be in the breast, and also the tumor that has disseminated either locally or at distant sites. There's obviously a change in the biological characteristics of that tumor in terms of, for example, their ability of these tumor cells to move and survive in circulation, and then also resite and survive in different areas. And so we're kind of interested in exploring these biological differences. And then obviously recently, over the last number of years, people have become very interested looking at more closely within the tumor itself and looking at heterogeneity in terms of different cell populations and different molecular subsets within those tumors. And this is an important context in terms of treatment. For example, we could have a certain subpopulation which is inherently resistant to particular types of treatment, and it could be present at a very low level or low proportion within the tumor mass. And while we may get overtly a good treatment response in the bulk of the tumor, that residual low subpopulation may actually survive and then go on to contribute to remission or relapse of the tumor over time. So that's a context. We have diversity at multiple different levels, either between the patient, between the tumor, or within the tumor. And what we've seen is that we've had a lot of really fantastic advances over the last number of years. We have great information at a molecular level. And really what we want to do now is harness this new data to redefine how we treat breast cancer with the ultimate goal of more precise therapy. And again, we have an issue in terms of the complexity of types of data that we're dealing with. We're, for example, working with gene expression data, proteomic data, mutation data. Really what we want to do is combine this information in a particular way that we can actually add value to the current treatment paradigms which are already present. And this derives this new concept of systems medicine, where we actually can integrate data at multiple levels to really address this clinical need. Another key context behind the centre is really tackle some of the key factors which are hampering progress in breast cancer research. And again, it's one of the areas that was highlighted in that gap analysis document, was really, I'll come back to that in a subsequent slide. So what I'm indicating here is really this concept of leveraging data to personalise therapy. So as I indicated, the centre has three main components. We have a translational component, a clinical component, and a population-based component, all gravitating around a research core. One thing to note that when I talk about this, again, this cooperative oncology group, ICORG, which is carrying out clinical trials within Ireland, they've essentially recruited over the last 15 years approximately 7,000 cancer patients in clinical trials. Of those, a very significant proportion of those are within the area of breast cancer. So we have a rich resource of clinical trial material that we can actually get access to carry out research, which I'll come back to later on. So again, when we look at dissecting breast cancer at different levels, we obviously have an interest in looking at the gene alterations at, for example, a mutational level, expression level, or protein expression differences. But it's important to be able to map this type of molecular data with robust clinical information, but also combine this or marry this with what's seen at a population-based level. So it's important that when we generate data that we can actually integrate it into a centralized database, so where we can actually look at these different aspects. Again, from this, following this data integration, we want to come up with new diagnostic options and therapeutic options that then can be subsequently validated within our patient populations. I mentioned this concept earlier, which is really one of the barriers to breast cancer research, but also other forms of cancer research, is really access to relevant biological materials, particularly specimens from patients which have relapsed on disease. You know, sometimes it's quite difficult to get access to metastatic material, fresh metastatic material, where you can actually carry out robust investigation. So I've indicated here on the schematic is really looking at the various different stages that a female with breast cancer could go through. For example, you may have healthy women, which can be profiled into low or high risk. But really, what we focus on is really in the context of patients when they present with primary cancer. Can we, for example, assess the molecular profile of that tumour to determine what's the most appropriate treatment? A person may have initially a remission, but unfortunately may relapse or even initially present with metastatic disease. And this is where it becomes very important to be able to compare the primary tumour versus the metastatic lesions. And this is one of the goals behind one of our work packages within Breast Predict. As part of our centre, and this brings in the population-based level aspect, is that we're really interested in looking at the impact of lifestyle factors, but also exposure to other medicines, for other commonly prescribed medicines, for example, aspirins and statins, in terms of the impact on breast cancer outcome and response to treatment. And so we actually have ethical approval now, and we're rolling this out on an all-Ireland basis, all-country basis, to really gain this type of information from all breast cancer patients within Ireland. So we have 8 major cancer treating centres within Ireland. All of these are on board for this particular programme. So some of the questions that we're answering in our research programs is, as I mentioned, that we know that a significant proportion of women actually take various different prescribed drugs prior to their breast cancer diagnosis. And we weren't really interested in what is the impact of those drug exposures on the breast cancer that they actually eventually get. And so what's really the impact of commonly prescribed drugs on outcome and response to treatment. Another key interest within the centre is really looking at how do tumours evolve, breast cancers evolve over time in response to treatment. And again, one of the kind of key interests out there, again, as I mentioned, we have a huge arsenal out there of different therapeutic options in breast cancer. We have various different classical chemotherapies, but also new and exciting targeted agents. And the key question is, how do we effectively combine these together to get maximal impact? And we're interested really in combining both in silico and network modeling approaches to come up with optimal combination strategies, but also to use very sophisticated in vitro and in vivo models to actually come up with optimal combinations that will actually then be translated and tested in a human context. So we have 5 main goals within our centre. The first is really to provide a consolidated national breast cancer biobank and database, and a lot of work has already taken place by a number of different investigators within Breast Predict to generate a robust biobank. But we want to layer around that a really sophisticated databasing infrastructure that we can actually pull in data from multiple different aspects. And then once this is our kind of core of materials and information in the context of breast cancer within Ireland. And around this, we have 4 research-oriented work packages which are asking specific questions. And one of these is looking at the impact of aspirins and statins taken prior to breast cancer diagnosis in terms of the impact on outcome. Another question is looking at, another work package is looking at how the tumours evolve in terms of response to endocrine therapy and HER2 targeted therapies. Again, as I mentioned, we have a work package focused on modeling the best combinations of anti-cancer therapies, particularly combinations of targeted agents. And then we also have a work package focused on diagnostics. I'm going to elaborate on each of these programs in the subsequent slides. So the first work package, which is a central consolidation work package, it's really about building a comprehensive infrastructure and also capacity building. This is led by Professor Leonie Young, who's based in the Royal College of Surgeons in Ireland. And Professor Young has a huge amount of expertise in really building a robust biobanks, particularly at the College of Surgeons, but also she has actually made a lot of efforts now to expand beyond that to other centres around Ireland. And the deputy lead for this activity is Professor Michael Curran, who's a very prominent surgeon based in Galway in the west of Ireland, and has also kind of led similar efforts to establish biobanking activities within that region of Ireland. And again, they're joined by other members within Work Package 1, really to provide a very comprehensive platform for investigation. So the various different subcomponents of this activity is really to provide a resource, tissue and blood from breast cancer patients, both at primary diagnosis, but also importantly during treatment. So we're providing really a longitudinal resource that we can actually use for downstream experimentation. Another key feature behind it is there's obviously big interest now in the cancer community to working with more relevant in vivo models, particularly the use of patient-derived xenograft models. And we're using two complementary models and generating a variety of these as part of the center. For example, we're taking primary tumor material, but also material from metastatic lesions to create PDX models, but also looking at combinations of tumor cells, but also patient-derived stromal cells, and looking at the impact of these on within in vivo models. Nicely, around that, we also have very comprehensive molecular imaging platforms to actually profile what happens to these tumors in terms of their growth rates, their metastatic potential, but also their response to treatment. As I mentioned, we have a component around building a national breast cancer database. And a key aspect behind this is being able to pull in data, for example, from the National Cancer Registry in Ireland, the national prescribing databases, drug prescribing databases, breast cancer screening data, but also data that's resident within various different pathology departments. And one kind of, I suppose, more blue skies aspect behind this program is obviously we have an important resource in terms of emerging molecular data that's going to be generated in subsequent work packages, but also we have this clinical data that's going to be present within that patient set. And there's really an opportunity to combine this information into developing virtual patient models that can be used down the line. So work package 2 is focused on uncovering pathways from population drug exposures to breast cancer outcomes. And this particular work package is led by Professor Kathleen Bennett, who's based in Trinity College Dublin. And Kathleen is an acknowledged pharmaco-epidemiologist. And the deputy lead for this program is Dr. Annette Byrne from the Royal College of Surgeons in Ireland. And Dr. Byrne is a leader in the area of modelling of cancer in vivo within small animal models. And so one of the components is that we really want to focus on this link that is seen in terms of exposure pre-diagnostically, or patients, breast cancer patients, before they get their diagnosis of breast cancer, looking at the potential impact of aspirin intake in terms of their outcome. And so approximately we predict about 1/4 of patients, breast cancer patients, would have taken low-dose aspirin, which is actually prescription only within Ireland, prior to their breast cancer diagnosis. And investigators within the team have actually published a paper in Cancer Research, which has shown that aspirin seems to have a protective impact in terms of reducing the aggressive characteristics of the breast cancer that's presented when patients actually take low-dose aspirin prior to their diagnosis. So one thing that we're kind of keen to do is really expand beyond this study and actually look at, for example, the impact of known cancer-driving mutations, particularly looking at the impact of PI3 kinase mutations on this effect, and also look at this pathway at a bit more mechanistic level, looking, for example, at members of the COX-2 family and prostaglandin pathway, to try to understand why we're seeing this potentially protective effect. Alongside this kind of, I suppose, molecular pathology studies, we're also trying to dissect the mechanism of action of aspirin within more relevant in vivo models. And one of the concepts is that aspirin may have an impact on lymphoangiogenesis. And so one of the kind of key goals within this work package is to establish robust animal models combined with imaging approaches to look at the impact of aspirin on lymphoangiogenesis. And again, as I mentioned, this particular work package is focused on both aspirin and statins, and we have some data from our lab showing that when we look at the expression of one of the drug targets for statins, HMG-CoA reductase, at a protein expression level within breast tumors, high expression of that particular enzyme, this drug target for statins, seems to be associated with a modified response to endocrine therapy. And so we're very interested in really seeing if this is really relevant, this observation we've seen in breast cancer tissues, is relevant in a population-based level, and that's an area that we're actually exploring within the centre model. Work package 3 is looking at the impact of or looking at the evolution of tumours during therapy, particularly in the context of endocrine therapy and therapies which target the HER2 protein. This particular activity is led by Professor Brian Hennessy, who's a medical oncologist based in the Royal College of Surgeons in Ireland, and the deputy lead is Professor Des Higgins, who is a kind of world famous bioinformatician who developed, for example, the sequence alignment program Clustal. So in a variety of different kind of concept studies, what we're doing is leveraging the resources that we have within the this ICORD, or clinical trials infrastructure within Ireland, where we can actually look at patients in a longitudinal manner, looking at the how they evolve during treatment at a molecular level. And so what we've done is we've identified a number of different clinical trials within the portfolio of ICORD, where we have patient samples at various different intervals during treatment. For example, before neoadjuvant treatment, at the point of surgery, and at the point of relapse, etc. And what we're doing is we've gotten access to those materials, and we're performing multi-pronged omic assessment. For example, looking at whole exome changes at a mutational level within the DNA, looking at transcriptomic alterations using RNA sequencing, and then also looking at protein expression and signaling changes using reverse phase protein array technology. And again, we have a lot of expertise in really kind of marrying these these various different types of data sets together, be they DNA alterations, RNA alterations, or protein expression alterations. And we want to really come up with really, I suppose, novel pathway alterations which maybe explain, for example, either preferential sensitivity to these types of treatments or resistance or emerging resistance. So following these discovery studies, which are still ongoing, we also have a program of downstream extensive validation, where we're going to look at really extrapolating these findings on a much broader set of patient samples, really to bring these observations closer to clinical implementation. Work package 4 is really focused on this concept of combining novel agents together to really come up with novel therapy combinations based on a more sophisticated understanding of the biology of breast cancer, rather than just some empirical mishmash of combinations or drugs that you might use. This activity is led by a noted medical oncologist in Ireland, Professor John Crown, who's based in University College Dublin and Dublin City University, as well as St. Vincent's University Hospital. And the deputy lead is Professor Walter Colch, who's director of the Conway Institute and also Systems Biology Ireland. So this particular work package has three main components, which I'm going to outline here. The first is really to develop and utilize a computational modeling framework where we can actually link this omic data, for example, data coming from this work package 3, which I previously mentioned, but also external data that's already out there. There's a slew of omic data and other similar data set out there in the breast cancer context that we can actually leverage, with various different signal transduction models which actually have been developed within the context of Systems Biology Ireland. And the idea is really to use this information to really come up at an in silico level with novel druggable nodes, which can then be subsequently tested experimentally within the lab using in vitro and then following that in vivo models using both gene knockdown technologies but also therapeutic agents. Alongside this computational modeling framework, we also have a plan and we have ongoing activities to really perform robust functional screening, where we use various different combinations of drugs and libraries of siRNAs or short therapeutic nuclear RNA to knock down genes of interest in essentially synthetic lethality screens in various different model systems to come up with novel combinations. So we have both a modeling concept and also a functional screening aspect. So along these more kind of generic broad-based approaches, we have some very targeted projects where we're actually going with already kind of known druggable entities and looking at specific combinations. We're also looking at targeting mutant p53 using specific agents which can actually change the properties of mutant p53, convert it into a more wild-type form. And then also we have a specific basic research program looking at epigenetic silencing of the p16 gene. So the last work package is looking at kind of diagnostic implications. This work is led by Professor Joachim Prenn, who is based in the College of Surgeons, and also the deputy lead is Professor Lorraine O'Driscoll, who's a kind of leading investigator, emerging investigator within Trinity College Dublin. And so this program has 4 different aspects. The first actually is looking at apoptotic signatures. So Professor Prenn's group has actually spent a lot of work really modeling various different components within the apoptosis signaling pathway using a systems modeling approach. And he has shown in a colorectal cancer context by measuring essentially a small number of components within the apoptosis signaling pathway, he can actually come up with his group can actually come up with a predictor of inherent chemosensitivity or chemoresistance. So he's shown this concept using clinical tissues from colorectal cancer patients, and one of the goals behind the center is really to validate a similar concept or a similar approach within breast cancer. Professor O'Driscoll has done a lot of work really on looking at circulating nucleic acids, particularly microRNAs, but also more conventional messenger RNAs within cancer patients. This work has also been done by Professor Kerrin and Dr. Roshan O'Dwyer in Galway. And there's a big interest in really using these types of circulating nucleic acids, either free circulating nucleic acids or ones which are, for example, bound within exosomes as biomarkers of response to treatment. Professor Leonie Young in the College of Surgeons has a huge wealth of expertise in looking at molecular determinants of response to endocrine therapy. And she has a particular program where she's identified a very interesting putative marker of endocrine resistance that she is bringing forward at a further level of clinical validation within the center. And the last program here within this work package is led by Professor Rosemary O'Connor in University College Cork, and she has really a huge amount of expertise in IGF signaling, and she's identified really a very interesting signature which may hold potential in the breast cancer context. We're working very closely with her to really translate this observation from the basic science end into a more clinical context. So as I mentioned, we've really been in existence for a year. We've really been very successful, I think, in establishing this network of both 20 leading investigators in the breast cancer field in Ireland. The actual funding behind the Irish Cancer Society, from the Irish Cancer Society, has really provided an additional over 30 individuals, both PhD students, postdoctoral fellows, research nurses, and other staff. So an extra over 30 individuals combined with those 20 investigators really create a team of over 50 people working towards a common agenda to tackle specific issues relating to breast cancer. Within that year, we've published 12 research articles within prominent cancer-focused journals. We've also published various different review articles. We've organized a variety of we've established new collaborations, and we're actually open for collaboration with different partners around the world. We've also organized various different national and discrete seminars and also bigger workshops. We've also enrolled a number of different patients within various different clinical trials, and we also have really greatly accelerated the number of specimens that can be used for translational research. In addition to these kind of core activities, we've also been able to leverage additional funding from other organizations, both nationally and internationally. I suppose a key part of the centre is really, because we receive funding from a charity, it's very important that we communicate our work to the public, because they're providing the resources for our research. And so we've actually spent a lot of effort really kind of publicizing our efforts and communicating these to the scientific advances to the public. So we have a Breast Predict website, which I'll indicate at the end of my presentation, and I would encourage you to look at that if you're interested in more details. This is just an indication of the types of workshops we've organized over the last year. And again, as I mentioned, we've published these a variety of different articles over the last year. I'm going to highlight just a couple of these just to provide a bit more detail in terms of research. So the first really refers to the activity within work package 2, and this is from the work of Professor Bennett and Dr. Ian Barron. And this is really looking at pre-diagnostic aspirin use and mining data from the National Cancer Registry in Ireland to identify women who have taken low-dose aspirin prior to breast cancer diagnosis, and then looking at impact of that exposure to low-dose aspirin on their outcome and also other different parameters relevant to breast cancer. And as you can see here, that if you look at all breast cancers, there seems to be a significant, if modest, impact in terms of aspirin on survival. This is looking at breast cancer specific survival across all breast cancer patients. But if you look more specifically at patients which are node negative, you have a very significant impact in terms of a potentially protective effect of aspirin, or a modifying effect of aspirin when taken pre-diagnosis to breast cancer. Dr. O'Driscoll published a paper recently in Cancer Research on this very interesting protein neuromedian mu, and they identified this protein really in initially in cell line models which acquire resistance to HER2 targeted agents. They showed that this factor is increased in cell lines which are resistant to those types of agents. They also showed, by using a particular integrated resource that we developed in our lab called BreastMark, where we really assemble gene expression data from publicly available gene expression data from breast tumors, where we have survival data from the patients. We have data, for example, close to 4,000 individuals. When we look at expression of neuromedian mu within all breast cancer subtypes, we show that high expression of neuromedian mu is associated with poor outcome at a very significant level. Then when Dr. O'Driscoll's group actually reduced the levels of this particular protein within particular cell line models of breast cancer by using a short hairpin looped RNA approach, by knocking down expression of that particular factor, she showed that you could actually decrease both primary tumor growth but also metastatic potential. Another key factor is that when you suppress this particular factor, you can actually resensitize these what were resistant cells to HER2 targeted therapies. So the last paper I'm going to showcase is really work from Professor Leonie Young and her group in oncogene. And this really comes to a very interesting concept that we know that, for example, obviously there's two main treatment options used in breast cancer. As I mentioned previously, we've got endocrine therapy targeting patients which express the estrogen receptor, or HER2 targeted therapies which overexpress the HER2 protein. And again, there seems to be some crosstalk between these two different pathways. And this paper really tries to explain this crosstalk in a bit more detail. And from this paper, it's seen that patients which are HER2 positive, which have a functional ER pathway, seem to have poor outcome when treated with trastuzumab. And Professor Young's group really provided a pretty nice or kind of mechanistic concept behind this. And it's known that, for example, if patients are which have an overexpression of the HER2 receptor are treated with trastuzumab, you get modification of PI3 kinase signaling, and you get some kind of signaling from PI3 kinase to the MYC gene. And what we've seen is that there's an interplay, there's an effect, there's also an effect in terms of when you have an impact in terms of PI3 kinase signaling, there seems to be a induction of interaction between the estrogen receptor and this co-integrator protein CBP, which can actually be contribute to enhanced transcriptional activity. So in summary, when you treat with these types of agents, such as these HER2 targeting agents like trastuzumab, it may actually inadvertently lead to increased activity in terms of the estrogen receptor, and therefore contribute to some kind of crosstalk scenarios. So where are we going forward with the center? So again, as I mentioned here, these are the various different goals within our center. But really our vision is really to develop our a vision beyond our 5-year timeline. As part of that, we're really actively focused on leveraging funding from independent sources to both accelerate progress, but also put in place a model for beyond our program, our current program. But also, more importantly, really to make personalized medicine a reality for breast cancer patients. So again, if you're actually interested in more details behind this program, I would encourage you to look at this website, and you can also find our contact details on the website. So I certainly have time for questions if there are some. I'm not sure if I was going to have a look, open up the Q&A box. I'll give a minute or two if there's any questions. If not, I can close it. So I don't think there's any questions. So I think we can probably close the session.